Hello, my name's Rob Durham,
And I'm John Chappell.
and you're listening to another episode of the Gyrolab Spin. On today's episode, we're going to talk about how the Gyrolab can be used to increase productivity in cell and gene therapy bioprocessing.
Cell and gene therapy is very much a growing and exciting area. It's producing very effective and fast treatment, so it's very exciting to be involved in this research.
That's right, John. And part of the drive from our side is that the recent approval of a lot of gene therapies has made this a very hot area, and it's really contributed to problems in limited manufacturing capabilities, capacity. And so I think one of the places that the Gyros helps is to really streamline some of the processes involved in key quality attributes of the therapeutic products.
Saving of sample volume and speed is a key to this support, really.
Yeah, and so anytime you can sort of automate a manual method and it reduces some of the art associated with doing an immunoassay, it really helps to facilitate reliable, consistent, and faster analytical methods. From a gene therapy perspective, one of the key tools that are used are these lentiviral vectors, which are based on an HIV-1 virus. The nice thing about these is that they can actually insert a gene into a cell, and that genomic insertion means it's going to be a permanent fix.
Yeah, no, lentiviruses bind well with T cells, so it's a natural way of modifying the immune system. So a lot of immunotherapies are using this CAR T cell approach, where you take the patient's own T cells and then you modify them,
Yeah.
giving them the ability to attack cancer cells. It's a very exciting bit of research, really.
Yeah, and the other thing that's important, in order to create the viral vectors in the first place, we need a cell line, and so there's a bioprocess component to this workflow as well. So often we're using the HEC293 cell lines to produce the viral vector, and so from a Gyros perspective, then we provide tools to measure the viral vector, a titer, physical titer, as well as tools and assays to measure the host cell products or contaminants that might be left behind.
So, as mentioned, we have a Gyrolab P24 kit for lentivirus quantitation. So this is a kit which uses a capture and detection antibody that bind to P24, and the kit contains all the reagents to fully automate this method, and it uses very low sample volume and has really high throughput.
So for lentiviral vector quantitation, we can do this in a couple of different ways. The first is a functional or infectious titer, and this is done usually using a cell culture system. It takes quite a long time. It's not practical in all senses, but it gives you a measure of how well the gene is transduced into the cell line. The more common way is a physical titer, where we're actually just measuring the number of particles. This is a more accurate endpoint, not as time consuming, and is sufficient in most instances. So in this case, the assay principle is to measure the P24 particle, which is a surface protein on the lentiviral envelope.
So as mentioned, P24 is used to determine the titer in lentiviral production. It is a component of the particle, and in cell culture supernatants, P24 can be present free in solution. But what we do also do is we use detergent to actually break up the capsid and release the P24. So you'll find as part of the sample preparation that Triton is used to actually break up the capsid, and this is quite a normal part of the sample preparation procedure.
Right, and then so what you end up with then is usually a ratio of between free virus and associated P24. Both pre and post
exactly.
the detergent treatment.
Yeah.
So here you can see an example where we're comparing the effect of the detergent treatment on standard, which you'd expect has no impact because it's not a full viral particle, versus a sample where the addition of the detergent breaks up the viral particle, and we see an increase in the measurement of P24 that's free in the solution.
So here we can see some dilutional linearity data. So this is demonstrating the performance of the method. So we've taken some samples, and you can see samples with and without Triton. And as you would expect, when the sample's treated with Triton, you get an increase in the P24 levels, and you can correlate before and after treatment. And you can see it approximately for one sample increases by around 4 times, and in the other sample it's around 9 to 10 times increase in the P24 levels.
Yeah, and but the I think the point here too is that you're getting good dilutional linearity both before and after, so you're confident in the values that we're getting
it correlates that the method is giving good performance and what you're measuring is accurate. So when we look at the Gyrolab assay range, it's a very wide dynamic range. Gyrolab uses laser-induced fluorescent detection, so that normally gives a large dynamic range, and in this case it's between 0.2 and 1,000 nanogram per mil, which is much bigger than an ELISA method, for example. So the advantage of this is that we're more likely to get the samples in the range first time, and particularly for bioprocess, where dilutional linearity and spike recovery are a very important part of the analytical process. It's more likely with that we're going to be able to measure things first time. So also the method performance, it shows great precision and accuracy, showing that the assay is very reproducible, and you can also see 6 standard curves, and you can see how reproducible they are over time.
Yeah, it's one of the nice things about this automated method is that you get good solid reproducibility, no worries about operator to operator variability as well. So the gyro system can also be used for titer of the adeno-associated viral vectors. So AAV, unlike the lentiviral vectors, inserts a DNA that becomes a stable episome that resides in the nucleus, but doesn't become incorporated into the host genome. So this has some advantages from a safety standpoint, as well as but limitations are around the effect can go away over time.
Yeah, I think the good thing about AAV as well is that you can target specific tissues and organs, different isotypes, different serotypes, sorry, depending on what disease that you're trying to treat. So we're seeing pretty much most of the serotypes people are developing therapies with, so depending on the treatment. So and it's obviously a very growing area currently.
And so there's a number, at least 10 different serotypes, and I think more are being created all the time. But the other factor here is that pre-existing antibodies towards various serotypes can limit the potential efficacy in patients. So it's one of the considerations for patient selection as well.
Yeah, I mean, AAV is a non-disease causing virus in humans, and most people are exposed to it, particularly in childhood. So it's likely that there will be some pre-existing antibodies. So it becomes a very important part of the kind of inclusion criteria of a study, whether someone has pre-existing antibodies, and if they have high pre-existing antibodies, they may not actually have the therapy.
So as we mentioned, one of the challenges then is to have some sort of a serotype specific assay. One of the problems here is if you have to have a different assay for each of the 10 serotypes, well, then that makes it sort of complicating. So what we have here is a rather generic method that will cover a broader range of the serotypes. So we're using the Capture Select anti-AABX ligand to bind to the surface proteins on the lentiviral vectors. And this will give us a good assay that provides broad dynamic range and also binds to a wide range of serotypes. For this one, we're able to measure serotypes 1 through 10, except for AAV9, I think, is the one that is left out. It
Yeah,
doesn't bind well.
and we have a separate method for AAV9, as we'll show in the next upcoming slides. But this assay is performing very well in the field. I think customers in general are very happy with this assay performance, and it's being used for many different serotypes. They use their own standard, their own viral vector to actually calibrate the assay.
Right.
And I think we're about to see that each serotype gives a slightly different analytical range. So here we can see standard curves done with different serotypes from 1 to 8 and AAV10, and you can see that the analytical range slightly differ, but they all cross-react very well with the assay, and we can demonstrate assay performance with every serotype, and obviously you would validate the assay with your specific serotype that you want to use. So it's a very flexible and open assay, and it gives really good performance.
Yeah, but I think sometimes there's concern because you see that AAV8 curve shifted to the right, but still it's well within the analytical range typically required for a titer assay.
Yeah, and I mean the good thing is it's a wide dynamic range, and it gives obviously uses low volume, so it's very easy to actually measure samples using this assay. Here we see some performance data generated using AAV2. So this shows precision and accuracy using quality control samples, and we can see great performance. All CVs are less than 10%, and between runs less than 11%. And we get really good accuracy, within 10%. So overall, the assay performs very well.
Yeah, I think when you have labeled reagents, a kit, and automated system, it really streamlines the process.
So when we look at the AAVX kit performance compared to ELISA, you can see that the dynamic range for the Gyrolab method is much larger, over 3 logs. And you can see the ELISA is only about 1.5 logs. The sensitivity is very similar between the Gyrolab and the ELISA method, but what we've seen from customers is that the upper range is really important, particularly in the production process, when they're starting for quite high viral particle titers. So getting samples into range is much easier with the Gyrolab, and particularly with the samples being so precious during the production process, this becomes a much more viable method to actually be incorporated into the production process.
Yeah, I think oftentimes it's that getting your diluted sample onto the curve much more challenging if you only have a 1.5 log target versus a nice broad range in the Gyros assay.
Yeah, and you end up, if you miss the range, you end up having to repeat the sample and actually use even more sample trying to repeat it to get onto the range. So we're avoiding that part.
So let's go now to talk about the HEC-293 host cell protein solution for Gyrolab. In this case, we've partnered with Cygnus, who's one of the leading providers of ELISA kits for the host cell protein market. And in this partnership with Cygnus, they provide the reagents that are the same reagents that they use in their 3rd generation HEC-293 ELISA kit. And we combine that with a toolbox from Gyros, which includes the CD, the BioAppy 1000 HC CD, that can be used to develop your own HEC-293 assay.
Yeah, I mean HEC-293, as already mentioned, is one of the main cell that is used in viral production. So this kit is mainly used in cell and gene therapy, but it is used by other bioprocess projects. But the reagents are generic, is that they're designed to be wide spectrum and essentially will measure a whole host of host cell proteins that can be part of the bioprocess purification process. The antibodies are polyclonals that have been generated in goats and rabbits. So a polyclonal will give you a wide response, will react with many proteins in the HCP mixture. So it's a very, it gives really good performance, and it really does work very well.
Yeah, I think one of the things that we want to we're always concerned about is when somebody is using one method on one platform and tries to change the platform to something more automated such as ours, then they're concerned about what the comparability is going to be, right?
Yeah, and it's sometimes they look at the absolute values between maybe an ELISA and a Gyrolab method, for example, and sometimes the results will be different. But the important thing in the purification process is that you're reducing the HCP levels, so the absolute quantification isn't critical.
Yeah.
It's just about the ability to show that you're removing the contaminant. That's important.
So as we mentioned already, the benefits of the 293 solution is that it allows you to automate the workflow from a manual method, gives broad dynamic range, and really pretty fast turnaround relative to a manual ELISA method, and the results are comparable to the typical ELISA.
Yeah. So the advantage of like the toolbox is that it uses a high capacity CD.
Right.
The high capacity CD is very good when you're using polyclonals for capture.
Yeah.
So it gives a very high binding capacity. So it makes it very sensitive for measuring a wide variety of the host cell proteins. The good thing about this toolbox as well as being able to be used with the Cygnus reagents, you can also use the same toolbox if you want to develop your own process-specific HCP method.
Right.
And we do get a lot of requests in our assay development services for people to develop, and we help them develop their product-specific HCP assays.
Right. So the Cygnus is a great tool for a generic, non-specific process contaminants, but the same toolbox works equally well for process-specific.
So again, with the Gyrolab method, we have a wide dynamic range. So the working range is from 4 to 8,000 nanogram per mil, and it gives very good performance. Here we're seeing precision and accuracy data with QCs, and we're seeing acceptable performance. This data was actually generated by Cygnus Technologies.
So again, here in some more data that was generated by Cygnus, you can see a typical standard curve from a run. There's duplicate measures there right on top of each other, performance that you'd expect. And again, from very low 4 nanogram per mil standard all the way up to 8,000, you can see very good performance of the assay.
So a very important parameter for bioprocess assay sort of measurement and also validation, and to demonstrate performance, to perform dilutional linearity of samples to show that you're not getting any bias in the measurement. And you can see here data are generated from 13 samples, and it shows very consistent performance over dilution, which is really to show that the method is working properly.
Yeah, I think when you have a method that's really semi-quantitative like this, it's really critical to show that within sample dilution looks good. Sometimes they'll even spike in standard to make sure that you're getting dilution linearity.
Yeah, they do both. So you do
spikes.
spike, yeah, you do with and without spike, and then that really shows that the method's given an accurate measurement.
And I think this highlights another advantage to using the Gyrolab in this case is that we've built in some dilution linearity spiked sample mapping tools into the assay setup and analysis, which has really been useful from a user standpoint.
Yeah, so you can actually design the sample dilution in software, and then it will present in the graphs and show you whether you've got a linear response and show what samples are within acceptance criteria. So it's a very useful tool.
Yeah.
As mentioned, the Gyrolab solution for HEC 293 is correlated against the ELISA. So in this case, they've taken samples and measured and compared it against the Cygnus HCP ELISA kit, and you can see that the Gyrolab results correlate very well, which gives you confidence, particularly if the customers are already using the ELISA in the lab. So it allows them to change methods or correlate with methods.
Exactly, and this kind of confidence, again, you're not going to get absolutely the same number. It's not that quantitative in terms of in like you would for a regulate or a bioanalysis measurement where you have a defined standard. Here it's a whole family of proteins, and so we're really working in a semi-quantitative assay, but still to get reliable, consistent performance across two different platforms gives one confidence that you can shift from one platform to the other.
Yeah, and as I said, the kind of the binding capacity of the column and the Gyrolab is much higher than ELISA, so there is potential that we can measure other proteins that may be missed by the ELISA. So for some samples, you maybe get some differences, but as I said, the important thing is that during the production process, you're showing a reduction in the HCP,
right,
as part of the analytical process.
Well, in today's talk, we've talked a bit about how immunoassays are really an important tool in the cell and gene therapy area, and to automate and speed the process of manufacture of these critical therapies that are delivering really huge benefits to patients. We've shown that I think having an automated method helps to establish titer and impurity levels. And the Gyrolab really represents a robust alternative to manual methods.
Yeah, and the large dynamic range makes it easier to get samples in range first time, so it does conserve precious samples and reagents, which is very important. It means it also, particularly in analytical development, when they're screening many samples to kind of correlate with the production, it gives very fast results. Yeah. And I know from experience working with some customers that this like turnaround time can be critical, so they can provide the results back very quickly in the same day, and then that they can make critical decision about the production. So that speed is very important. And we've covered both areas in terms of AAV and lentivirus. The performance is very good, and the convenience off-the-shelf, ready-to-use kits, which particularly is useful when the assay moves into the quality control area, when we're seeing Gyrolab assays being adopted in QC from a regulatory perspective, which is important. Mm-hmm. We hope that the Gyrolab methods will become the gold standard for cell and gene therapy. Mm-hmm. Because it very much we operate very well in regulated environment. Our software is CFR 21 Part 11, and it does really fit into GMP areas. So, which is
right,
good.
Yeah. And the convenience of that off-the-shelf method, so you're not doing a lot of assay development. You don't have to maintain the reagents and all of the documentation that goes along with lot release, of critical reagents for supporting an assay throughout its life cycle. Having a generic off-the-shelf kit really provides a benefit there.
Yeah, but the other advantage is, yes, customers can run the kits and off-the-shelf kits, but they also can develop their own methods, things like leach ligands, for example, or even things like the gene product protein. Yep. So we're seeing people using the instrument not just for the bioprocess, but also other parts, and of course immunogenicity as well. Yeah. So there's a lot of people already presented immunogenicity data on the Gyrolab platform.
So I think that's really terrific, and it's an exciting area. So it's fun to be supporting this really dynamic drug development.
Yeah,
area.
It's very quick. Yeah, it's really drug development on steroids. They're very much trying to move things through quickly.
Absolutely. Well, that just about wraps up this episode. And as always, please hit the subscribe button if you want to be sure and get updates when we release new podcasts. And John and I are always happy to take your comments, feedback on this podcast or other topics that you'd like to hear about.
Yeah, please contact us, particularly for future podcasts. We're very much happy, we'd like to work with the community and provide material that's going to be useful to everybody. So please let us know if there's anything you would like us to discuss.
Well, that's goodbye from me.
And it's goodbye from him.